Okay, I'm gonna probably spend another 10 minutes on here because this is very important. This is a Punnett square. It's going to use the genotypes, means the letters, the gametes of the parents to reveal what alleles are the offsprings might get. I need you to write this down. I say this every semester, but still students, sometimes they cannot make the connection. The gametes are every combination. Basically what you put on here is the every combination that you can get for gametes. So what you put on top of this Punnett square or on the side is the every combination, every possible combination of the gametes. So I'm going to keep repeating this a few times today, but make sure you can make that connection. I'm not saying there are 2 eggs here for female gametes. I'm saying there is a possibility the egg might have this big G, another egg might have this little g, and these are 2 possibilities for that gamete. For the sperm cells, there is a possibility the sperm that's going to fertilize the egg is big G, and the sperm that might fertilize little g. So it can be either or, but these are 2 possibilities. So we want to consider all the possibilities when we do the Punnett square. So the gamete, again, I'm going to say it one more time, is every possible combination that you can get. So big G, little g for male and female. So the way you're going to do Punnett square, you're going to get the possible gametes, and for females, it doesn't matter where you put female or male. You can have female here or male here, and usually they write the uppercase letter first. It's just a little bit cleaner that way, but it's not required. So female gametes, here what we have is big G, little g. For male gametes, we have big G, little g. Then you bring it down. So you bring this big G down, big G from here, big G and little g here. So big G comes down here, little g comes down. And then you bring again big G down here, little g here, and then little g, little g. Is everybody following me so far? Yes, no? Please tell me, give me a response so I can, move on, or I can explain more.
Following.
Yes, okay. So, and then something that's called genotypic and phenotypic ratio. You're gonna have to get these. Now, I said genotypes are these letters. And then it says genotypic ratio is 1 to 1. What does that mean? You have 1 big G, you have 1 big G, little g, 1 big G, little g. So you have 2 big G, little g, and then you have 1 little g, little g. That gives you the ratio for these letters, 1 to 1. Then you also have to get the phenotypic ratio. That's what's expressed. When you look here, this is color coded, so it's easy to tell. But this big G is for yellow. And if it's with little g, still the dominant one's going to be expressed. So you have 3 yellow and then 1 green. So your phenotypic ratio is 3 to 1. So on the questions, I might say you are mating female gametes, heterozygous female. You have a heterozygous female and heterozygous male. What is the genotypic ratio for this? Or what is the phenotypic ratio for this? You're going to do the Punnett square, then you're going to give me the numbers. So just to make sure that you guys are kind of following me, let me give you something for practice. So help me maybe with this, some of these problems. I'm not going to do all of them, just to make sure that you guys kind of understand it. So here it says for each of genotypes, big A, big A, big A, or little A, little a, below determine which phenotype would be for purple flowers are dominant to white flowers. So in this case, we're saying big P is purple and then little p is white. So because it says purple flowers are dominant, that means big P is purple. And then little p is white. Now I'm gonna ask you guys, this is big P, is this going to be purple or white?
Purple.
Purple.
And next one?
Purple, yeah.
Purple.
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 This last one?
White.
White.
White. So this
White.
tells me you guys understand dominant and recessive. So you can do some more problems. This is on Canvas. You can practice. Now, let's look at this next one. So we're gonna look at So now this question is asking, tell whether it's homozygous dominant, heterozygous, or homozygous. So I'm gonna probably do a few of these. So what is this? Is this homozygous dominant, heterozygous, or homozygous recessive?
Homozygous.
Homozygous dominant.
So I'm gonna just
Homozygous, yeah.
be here. I'm not gonna write the whole thing out. What about this one?
Heterozygous.
Heterozygous.
Hetero. This one.
Homozygous.
Homozygous
recessive.
recessive.
And this one.
Heterozygous.
Homozygous.
Hetero. So you guys understand that as well. So let's move on. Number 3, it says female dog is heterozygous. Actually, before that, we need to read this. In dogs, the gene for fur color has 2 alleles. Means you have 2 versions of the gene. Dominant allele is big F, codes for gray. So when you do these problems, you kind of want to break it down. So you want to say big F, gray. And the recessive allele is black fur. So I'm gonna say little f, black fur. Now this is your question. Female dog is heterozygous. What does that mean? What does she have out of these 2 alleles? Or, what is the combination that she has of possible?
One dominant, one recessive.
One of each.
Dominant and a recessive.
So it's gonna be big F, little f. And then it says male dog is homozygous or recessive. So what's the combination you have here?
2 recessive.
2 little f's.
Little
Little
f.
f, little f.
Then it says figure out the phenotypes and genotypes of the possible puppies using a Punnett square. So you guys kind of understand this, or you want me to continue doing the Punnett square for this?
Can you do the Punnett squares?
Okay. Yeah.
So we could just see one.
Yeah. So I'm gonna put male dog here and female up top. Need a little bit more space. So for male, we had little f, so I'm gonna say little f. And then for female, I'm gonna put it female up here, because female's up there. So big F, little f. So big F, little f. This is little f, little f. So you're gonna bring big F down, little f, little f, little f, big F, little f, and then little f, little f. Now let's look at this Punnett square. I think it's hard to tell which one is little. So I'm gonna say yellow. Actually, let me do the other way around. I'm gonna say big F is yellow. You can tell easily. Okay. So what is the genotype for big F, big F, big F, little f? You can also write them in percentages. So let's look at big F, big F. Is there anything that has big F, big F? Both?
No.
No, so that's 0%. Big F, little f.
Half.
Half, 50%. Little f, little f.
Half.
50%.
50%. And then phenotypes, black fur and gray fur. Now we need to look at this.
2 and 2.
2 and 2. So you have big F, little f, big F, little f. So this is going to be gray. And then when you have little f, recessive, homozygous recessive, that's going to be black fur. So I'm going to say 50% and then 50%. So that's one example. There are multiple questions that you can practice. I'm not collecting this worksheet. It's on Canvas, but you can use this for practicing. So it is, so 100% of my students can do this if I set this up for them. If I put big G here, little g here, they can do the Punnett square, they can give me the genotypes and phenotypes. But sometimes students get confused when I give the word problem. So that's why you want to practice with the worksheet, because it has word problems, so you can set up Punnett squares. So once you're able to set them up, then you should be able to get the genotype and phenotype ratios. And one more time, I'm going to repeat this. The gametes are the possible combinations. So whatever is possible or to be passed on. Big G is possible. It might be in the egg that gets fertilized. Also maybe little g. Same thing for the sperm. I want to stop and ask if there are any questions on Punnett square.
So the ratio is just the same as the percentage that you just did, right?
Yeah, it's the same. You can either write it as a ratio 1-to-1, or you can write it as a percentages. If we were to write this as a percentage, you would say 25%, 50%, and 25%.
Okay.
Will we be going over this in lab next week?
Yes, we are. And I do have some announcements at the end for the lab today.
Thank you.
Okay, so in this example, female parent is heterozygous. For seed color is a cross with the male parent that's also heterozygous. This is the same example. We just went over this. This is just summarized for you. So that's what you're mating. This is your, if it was a word question, this would be the wording of it. Female parent, heterozygous, is mated with male that's also heterozygous. But here it's not telling the seed color because it's color coded right now. This is called a monohybrid cross. This is called a monohybrid cross. And the reason is because you're only looking at one gene. You're not looking at multiple genes, 2 genes. So this is a monohybrid cross. Dihybrid cross, you're looking at 2 genes. So today we are only going to work up to, monohybrid cross. We're not working on dihybrid. Next time is dihybrid. So mono means one gene. If it was a dihybrid, maybe you are looking at this gene for color and also maybe gene for height of the plant. Big T, little t maybe for the height. If you have one gene, you do a Punnett square, you're gonna have these 4 squares. If you have two genes, it's gonna get a lot more complicated. You're gonna have 16 squares for the Punnett square box. So it's gonna get a lot more complicated. So that's why we don't really, most of the time, we don't really go more than 2 genes. So 2 genes, the maximum that the biology classes will do. So this is called a monohybrid cross. Your next quiz is monohybrid cross. And since both parents are heterozygous for 1 gene, are being evaluated again. Monohybrid, 1 gene. Dihybrid, 2 genes. So if we were doing dihybrid, we'd be looking at more than the color, maybe the height, like I said. Genotypes, big G, little g, indicates that all diploid cells, including germ cells, in these parents are both dominant and recessive seed color alleles. So genotype, again, the letters. This showing that there's big G, little g, that means this is dominant, this is a recessive, and this is heterozygous. But you don't really always know the genotype unless it's homozygous or recessive. When the germ cells divide by meiosis, germ cells, you guys remember the term from last lecture, germ cells are referring to the gametes, female gametes, eggs, and then male gametes, sperm. And then they divide by meiosis, and then the alleles on those chromosomes are randomly distributed. This is called independent assortment. So independent assortment was, there were different alternatives, and we said it's one, for humans, it's 1 in 8 million. So that's what I'm talking about when I say independent assortment. There's a chance that this gene or this sperm cell is gonna get picked over the others, 1 in 8 million. And all those germ cells contain different DNA information, genetic information. A gamete from the female parent and a gamete from the male parent unite at fertilization, and then you have a zygote, and then it goes through mitosis. But meiosis is referring to these gametes. If both gametes carry dominant allele, if they both have dominant allele, then the offspring will have 2 dominant alleles. So when the independent assortment happens, a sperm that has a big G gets picked, and then female egg that also has a big G that gets picked randomly, then this is what the offspring is going to have. But if the sperm, the one that has big G, gets chosen randomly, then and little g for the female, then this is what the offspring is going to have. Offspring will be heterozygous, same thing here. But if when the independent assortment is happening, the sperm with the little g fertilizes the egg with the little g, then this is what the offspring is going to have, little g, little g. So again, going back to this, these are just the possible combinations or possible alleles that we put up here. It's not, I'm not saying there are two eggs. There's, you know, the egg with this, egg with that, that's getting fertilized. I'm just saying that these are the possible combinations of the gametes. The Punnett square is going to show the proportion of the offspring of phenotype and genotype. So once you do the Punnett square, it's going to tell you the genotypes, which are these letters, and then the phenotype, which is being expressed. And this slide is talking about this Punnett square. On average, 3 offspring will have yellow seed with for everyone with green seed. So 1 green, 3 yellow. That's the phenotype. That's what gets expressed, because this dominant allele is the one that's getting expressed for these 3. And then here is the only one that has the recessive allele that gets expressed. And then genotype, offspring will have genotype big G for every 2 with the big G, little g, and 1 with the little g, little g. So this is just talking about these numbers here, 1-to-1 ratio. So Punnett score is gonna allow you to determine the genotypes of the yellow pea plants. I'm gonna keep using pea plants because that's what we've been talking about. So here it says there are 2 plants here. One is with the green and then the other one is with yellow. Once the reproduction happens, this is what you get for the offspring. But here, same thing, you still have a green seed plant and pea plant, not seed plant, I've been saying it too much, and yellow pea plant, and once they get fertilized or pollen transfer happens, then this is what the result is. So how come you're mating, you know, the same thing, you're getting different results? So that's what we're gonna look at next. So if yellow seed color is dominant, why are some seeds green when a yellow seed plant is crossed with a green seed plant? So why this one is only giving you yellow, but here you have yellow and green both. So this is when you look at if it's homozygous dominant, heterozygous, which one is mating with which. So that's what you look at here. So if a cross between a yellow seed plant, now the yellow seed can be either big G or big G little g, because only dominant is getting expressed. And a green seed, that's only going to be little g, yields all yellow seeds. The yellow seed plant is homozygous dominant. So what this means, that means you don't know if this plant is this or this. Now to find out, you know the result for this plant. You know that little g is always green. Maybe it's a true breeding plant. So you do a Punnett score. You do a Punnett score with this possibility, big G, and little g. So that's what you do here if the plant is homozygous dominant. Then you do another Punnett score with this possibility, big G, and little g. So if you have a plant that has homozygous dominant, this is your result. Your result is 100% heterozygous and 100% yellow. That's what you have here. Now, if the parent is heterozygous, then you're gonna do the Punnett score. This is your result. You're gonna have 50% green and 50% yellow. This is for this possibility. So that's what you got here. You got 50-50. So if you know the genotype for this plant, and then you can do different possibilities. It can be either this one or that one, and then you do the Punnett score, and you get the offspring result, genotype and phenotype. Then you can go back and look at the offspring, and that's going to help you determine if it was this one or if it was this one. So Gregor Mendel, that's what he was doing when I said he was predicting. He was predicting this is what the offspring is going to be looking like, or this is the color that pea plant is going to have, or this is the height that it's going to have. So he was doing hypothesis and prediction, and he was taking notes. So he was able to predict as he got, better at it, that this is what the offspring is going to be. And once you find this out, or which type of plant it is, then you have a record of that. So basically here we can say for this part the yellow is big G, it's homozygous dominant. But if you have this result for the offspring, then we can say that it is heterozygous. So it's heterozygous here. So depending on which one you're, which possible gametes are there, you're getting different results for the offspring. So if the cross yields some green seeds, the yellow seed parent is heterozygous. So that's going to help you basically determine the parent. So you're kind of going backward, you're looking at the offspring and determining the parent after.